The examples that I'm going to show you over the course of the next few slides all aim at showing how global gene expression, so of course most of these slides are going to be DNA arrays, how we can learn about the cell cycle, periodic behaviors, as well as the pathways that are involved in particular pathologies, the cell circuitry and how that cell circuitry responds to the administration of particular drugs. We'll think about pathogenesis, that is to say how a disorder expresses itself through changes in gene expression. And we're also going to be thinking about, of course, pharmacological targets and the effects of drugs on gene expression. All right. I've taken a couple of figures from a very broad but very nice review to sort of give you an example of how we can use this microarray data to learn something new about the behavior, in this case, of an organism that is yeast. So in this case, we have a big array, 260,000 oligos printed on the slides, probing some 6,000 genes. So we have a really good coverage of the genome of yeast in this study. What we point out at the beginning, however, is in yeast, at the time of the study, only 60% of genes had known function. So I hope that you get that what I'm trying to show you in this particular slide is how we can use this array to build on this number. And what the authors did was use a so-called guilt by association method. And that in that they take the messages from different phases of the yeast cell cycle and try to correlate those with genes of known function. So we'll come back to this picture in a moment, but let's just think about the mechanics of the work. So at different phases of the cell cycle, mRNAs are reverse transcribed and biotinylated, right? That is to say, modified with a biotin, then hybridized with the array. So we take here's our array, and there's our oligo, and here's our reverse transcribed message, and it's hybridized, and we have its biotin, and there's its streptavidin related fluorophore. So we read expression of a gene, that is the presence of the mRNA of the gene, as an increase in fluorescence at that particular spot on the array. So here's the array, and we see that we're talking about different phases of the cell cycle, and we see, without focusing in particular on what these genes are, we see that their transcription is essentially shut off at M phase of the cell cycle. So the idea here is that we can take a gene and say that because it's expressed alongside a host of genes whose functions are known, we can say that the likelihood is that gene has a function in the same part of the cell cycle as those genes whose function we have assigned. So some 416 genes displayed periodicity, right? You can think about that in the context of the total number of genes that were probed. Let's see, some 60% of those are known cell cycle controllers, and then 25% of that 400, so 100 genes, seem to be spatially linked, and maybe this has something to do with local chromosomal organization, but spatially linked with those cell cycle controllers. Now this is a very complicated figure, and so let's just take a moment to talk a little bit more about it. Mostly what I want to point out is that this interior ring illustrates the proportion of the genes that are more or less constant in terms of their involvement over different portions of the cell cycle. And the main thing to point out, or the most noticeable thing to point out here, is the change in DNA synthesis genes as we go through different steps of the cell cycle. So the proportion of genes involved in that transition increases dramatically and changes dramatically as we step from one stage of the cell cycle to the next. So again, I think let me just reiterate that the point of showing you these data is to say that we can discover something new about the organization of an organism And we can do that by looking at a very large array, essentially all the genes of the cell, and to draw new conclusions. And I think the authors, when you look at this paper, are very cautious about the conclusions that they draw, but the point is still made that there's new information to be found about the cell cycle in this particular case through the use of this kind of technology. And again, that technology is fluorophore conjugated hybridization using reverse transcribed message bound to an array of in this case hundreds of thousands of different oligos covering most of the entire genome of yeast. In this slide, we're still not quite on to human cancers and human diseases yet, but I wanted to give you a look at the transcriptional regulation of Plasmodium, that is to say the organism that causes malaria, as an example of how broad the applications of these technologies can be, and two, in terms of the identification of potentially new drug targets. Malaria is a bit of an orphan disease in terms of the interest that it garners from the major drug companies, because of course though the population that suffers it is large, the amount of money to be had is rather small. The profit to be made is rather small. However, it's still a disease of intense interest, and there's a lot to be learned about how to treat the disorder, from understanding the changes in gene expression as determined through these sort of these array sort of technologies. So in this case, these workers restricted themselves to the intraerythrocytic portion of the organism's life cycle. As you're probably aware, malaria goes through, the Plasmodium goes through a very large number of steps in its life cycle through different hosts, but in this case we're talking about that stage inside that takes place inside the human erythrocytes. And they challenged some 7,000 oligos that they assembled on the array with message, reverse transcribed message, taken at 46 different time points over that one cycle, one portion of the organism's developmental cycle. So with that 7,400 oligo array, they cover a very large portion, over 80% of the open reading frames of the organism. So they have pretty good coverage of the genome. And what they see is a continuous cascade of transcriptional regulation moving through the genome over these different stages of the life cycle. So the way we've organized this is in terms of what stage, effectively time, but stage, we've organized it here, and we see that some genes go down in so red is a loss of expression, and green is an increase of expression as we move into different specific structural phases of the organism. Interestingly, this is not linked to chromosome structure. So that's just these interesting factoids pop up. In terms of results, which again we're not so concerned with in this particular study as much as we are in terms of generally how the technology is being applied, we see that invasion factors are strongly induced during the late schizont phase, and that's when the erythrocyte ruptures and the schizonts then escape from the erythrocyte and then can go on and invade other cells. In any case, what we see is this pattern of gene regulation across these, and this again, they're not spatially regulated, but in terms of who's involved, changes as we move through the cycle of the cell. And let's look in particular at what the data look like. I should point out, of course, that this is not a photograph of the array itself. This is a reorganization of the array data to reflect that sort of the linkage of function of these up and down regulated genes. In this slide, we see that we have 14 oligos represented, and those are covering the largest open reading frame of the Plasmodium. And we see that they're organized in terms of in terms of the cell cycle here. And we see again that there's that periodicity, that this open reading frame is shut down and is turned on, and then it's shut down in different phases of the cell cycle. We won't focus so much on that, but instead I want to focus on this technological detail in that there are two dyes that are involved here. And first, that there's there's a reference pool of message that's assayed, and those messages that are taken, it's basically an average over all of the cell cycle, and those samples taken over the entire cell cycle, and those are coupled with this cyanine dye, so that when we see hybridization and fluorescence, this particular yellow fluorescing dye is our sort of our reference, our standard. And then when we're looking at different stages of the cell cycle, that message is coupled with Cy5 as a red fluorescing dye. And so when we look at when we're quantifying the level of expression of the gene, what we're looking at is a ratio or log of the ratio of the sample to the reference. And of course, this is color coded according to whether the expression is down relative to the reference or up relative to the reference. But you see here where what we're referencing our expression levels at different stages of the cell cycle against. So again, we have a couple of different dyes that we can use. They have very different fluorescence characteristics, and one is our standard or our reference, and other is our target. Finally, in this slide, we can move away from non-human organisms and move towards discussion of how microarray data can teach us about cancer. In this slide, which happens to be showing data from a meta-analysis, this Rhodes paper from 2002 is a meta-analysis, and in a meta-analysis, the goal is to amalgamate several different, perhaps entirely unrelated studies in a statistically rigorous and meaningful way. The paper itself is focused on those statistical methods, but for our purposes, I'm much more interested in just focusing on their results. First, let's talk about the studies. These studies utilize 4 different studies utilizing some 100 patients, tumor tissue from 100 patients. These patients had benign, localized, or metastatic prostate cancer. And in the microarrays, they were assaying some 20,000 overlapping messages from these different patients. Now let's talk a little bit about prostate cancer. These molecules, putrescine, spermidine, spermine, these are all known to be found at elevated levels in prostate cancer patients. These are compounds that are known to be anti-apoptotic and to be involved in cell proliferation. So these are known to be associated with the development of tumors and are observed again at high levels in prostate cancer. Now, back to their results. So observation in the microarray studies, DNA biosynthesis genes are overexpressed, no big surprise. Also, perhaps no big surprise is that these polyamine biosynthetic genes, that is the genes that are involved in the biosynthesis of putrescine, spermidine, and spermine, are also overexpressed. And this is no surprise, given that these are the enzymes that are involved in the conversion of ornithine, which is a member of the urea cycle, obviously, into these cancer-causing or cancer-related endpoint molecules. Normally, an enzyme known as ornithine aminotransferase, referred to as OAT in the study. This is an enzyme that seems to be downregulated in prostate cancer. Normally, this enzyme might help shunt ornithine into a less antagonistic pathway down into proline, but in this case, it's downregulated. Now, interestingly, one of the go-to treatments for prostate cancer is a drug called DFMO. It's a modification. Here it is. Let's see, difluoro. Where's the methyl group? There's difluoro. Yeah, there's the DFM group, difluoromethyl ornidil. Ornidil is originally identified as a chemotherapeutic. It failed. It's still on the market for different indications, but now we have this modified version, difluoromethyl ornidil, that is a a suicide inhibitor of ornithine decarboxylase. So used in the treatment of prostate cancer. However, and it is known to decrease the concentration of putrescine. However, the other molecules that are of interest here, spermidine and spermine, are not, the concentrations of those are not decreased. So somehow or other, even though we can inactivate some ornithine decarboxylase, the ornithine is getting past that and into these other pathways. One interpretation of the microarray data might be that in the absence of ornithine aminotransferase, there's still less of an alternative pathway for ornithine, and so we still wind up generating these compounds, spermidine and spermine. And so the upshot is that our meta-analysis reveals an interesting signature for prostate cancer. That is, not only the sort of well understood or certainly not surprising upregulation of the polyamine biosynthesis, but also a downregulation of the proline biosynthetic pathway utilizing ornithine aminotransferase. So an interesting not only as a signature of the cancer, but also as an interpretation of the failure of a chemotherapeutic. All right. On to another example. In this case, we're talking about class discovery. So how can we define the gene expression profile that describes a particular tumor type, and can we use that definition to predict what the tumor type is based on gene expression of an undefined tumor? In this case, we're looking at the comparison and contrast of two types of leukemia. First is acute lymphoblastic, and the second is acute myeloid leukemias. Before I say anything else, though, let me say that there are two distinct treatment regimens for these two different tumor types, and that misassignment of the tumor can be problematic. One of the particular treatments for AML cancers is daunorubicin, which is a topoisomerase type 2 inhibitor. It's another intercalator, and it doesn't work very well. Although it is used to treat ALL occasionally, the treatment is less successful. So the point being that proper identification of the cancer type is critical in this kind of for proper treatment. So backing up then, let's talk about the study. And this is as similar as what we've already seen in terms of organization of microarray data. Of course, there's a cDNA generated array of some 7,000 different genes, biotinylated messenger RNA or mRNA converted to DNA, biotinylated, blah, 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 the same as what we've already discussed. Some 38 subjects that had either type of leukemia. And they arranged those based on again a normalized expression. In this case, we're looking at blue and red, and blue means down regulation relative to the control, and red means up regulation. And when they segregate these based on whatever genes they're looking at, which are not really relevant again to our interpretation of this, we see two distinct expression categories that really obviously blatantly allow us to segregate those two cancer types into two different categories. So 1,100 genes found to have statistically significant association. That allows us to discover a class. That is to say, we have a mechanism now that we've discovered that allows distinguishment between these two cancer types. And then they used the top 50 of these genes, the most strongly correlated of these, allows them to predict with very high success rates, right? So they took these 50, used that as a predictor for assessing a tumor that wasn't part of their study group, and then classifying that into one or the other, and they're successful with very high. So to reiterate, this kind of study shows us that one, we can logically assign a tumor to a particular class. 2, that we can, this allows us to properly treat the cancer. Again, referring to whether or not we use an AML focused chemotherapy versus an ALL focused therapy. And 3, we can avoid misassigning. And I'll turn to the paper itself to read what they say about that. So here's a quote, again from the Golub article, Science 1999. "We took the opportunity to apply our class predictor to a bone marrow sample from this patient. The classifier produced extremely low vote totals for both AML and ALL. Neither lymphoid nor myeloid specific genes were highly expressed, thus bringing into question the diagnosis of acute leukemia. Examination of expression profile revealed that genes more highly expressed relative to leukemias, blah, 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 we'll skip over that, and say independent cytogenetic analysis identified a translocation characteristic of muscle tumor, alveolar rhabdomyosarcoma." End quote. So the point was that not only are we able to properly assign the leukemia to its proper class, we can also avoid a complete failure of chemotherapy by finding the right type of cancer, period. So a much broader class assignment. All right. So not only can we more logically assign particular cancer to a category, we can also make some predictions about the way a tumor will respond to treatment. So in this study from PNAS 2000, we've categorized our microarray results here according to responder or non-responder or normal expression patterns. And just by glancing at this, again, without respect to what the 16,000 unique genes being assayed here are, we can see that there's some very clear behaviors that jump out at us. First of all, for the normal tissues, we see a very clear expression patterns that allow us to, we see these genes upregulated in the spleen, for example, these downregulated in the spleen, here and here. But what I'll focus you on instead is the somewhat subtler pattern, and of course I'm not going to go into the statistical analysis of this now, but the subtler pattern here of down and up regulation of genes that distinguishes the responders from the non-responders. So we don't see that pattern here. Maybe if I switch my colors again, you'll see that in this set, it's a qualitatively different expression pattern, even without paying close attention to in detail what the levels are. We see that there's qualitatively different behavior between the responders and the non-responders. Let me clear that. All right, in any case, back to what they did in this study. Let's see. This is follicular lymphoma, is the cancer type that we're dealing with, and what we're trying to make sense of is how the patients respond to rituximab therapy. Let's see, so 32,000 spots on the microarray covering 16,000 unique genes. Let's see, we have of the 24, we have all categories. We have complete responders, we have partial responders, we have no or minimal responders, and we say that the non-responders are categorized, even though the expression pattern that's not so obvious here. The non-responders are categorized with normal expression patterns, and the responders have a series of genes that get overexpressed. Some 27 different genes are getting overexpressed: cytokines, complement, TNF signaling. All of these are going to be standard sort of usual suspects, if you will, for tumorigenesis. And the lack of their expression suggests that then the rituximab would be a poor treatment for these individuals. So the upshot being that rituximab therapy deciding what patient gets assigned rituximab therapy could be based strictly on this sort of an analysis of gene expression in the tumor. Reminding you that